Welcome to the 30-minute case study webinar series. This is John Sankey. I'm an engineer for True Blue Technologies. True Blue is a sales engineering firm. We represent 4 different companies, Aestus, Tersus Environmental, TRS Group, and West Bay Instruments. This series this presentation is from a series, and the series focuses on the topic of managing complex sites with high-resolution site characterization and focused remediation. Today, we're going to be focusing on the high-resolution site characterization. Today, we're very pleased to have Dr. Todd Hallahan discuss on how to locate DNAPL and bioactivity at a former dry cleaner site. Just so you know, Todd is a professor and the Sun Chair of Hydrogeology at Oklahoma State University and serves as the Chief Technical Officer for Aestus. As the recent NGWA McElhaney lecturer, Todd gave talks all over the world. So Todd, thank you so much for presenting this case study today, and you now have the lead.
Thanks, John. And if I've got this managed conflict of interest in my life of being a professor at a university that has grad students and those normal things, and then the commercial side with being the Chief Technical Officer for Aestus. If you got any questions about that, a relationship and how that works, feel free to ask at some point and we'll go through that. We want to be respectful for your time. We've done this before now and asked and got feedback that some people wanted 30 minutes, some people want an hour, but the majority wanted it to be 30 minutes. So we're going to do about a 20-minute presentation with Q&A and be respectful of your time on that. If you want to stick around and keep asking questions, we're going to hang out, but we'll make sure that the talk is done by then, so that way if you've got to run to another meeting, you can get that done. Submit your questions via the chat bar, and we'll be recording this for you. And if you need CEUs, you'll get a certificate after the presentation. We've figured out how to make GoToWebinar do that, so that'll kick out and you'll get CEU certificates if you need one. We're doing Starbucks gift card giveaway, and we put them in a random generator and kick out who gets the prize. And if you submit questions, you can increase the number of chances you got in the bin. So, and you'll be notified by the end of the week or beginning of next week whenever we get that all done. So that's the way we're working this, and we'll get rolling here. So thinking about going into a dry cleaner with an injection remedy, you can have different types of impacts of the dry cleaner, but in this case, we're talking about PCE impacts. So basic solvents from a dry cleaning site impacting the ground, and going in with a bioremediation injection with a carbon source to get them to get the halogenators going and get them to break down the products. And then scanning that to see what happened after you're done, to see where the material went and whether you get bioactivity to grow. So we want to understand how to think about these processes from an electrical hydrogeology perspective, and think about what it means for us for our science in terms of how we deal with bioremedies. So we spent a long time working on protocols, and we had a real benefit for scanning first and then drilling, because the medical industry has been doing it a long time and the oil industry has been doing it a long time. But each one of these steps has got all sorts of components to it, and it takes a long time to work out optimizing these components and making sure steps are in place to ensure QA/QC all along the way. But the key thing is, very similar to medical, you're going to take a scan, but you got a biopsy, and you'll have questions that you go, well, we've looked at a lot of these, and I think these are the things we have, but you still have to biopsy to confirm things and determine the chemistry you're working with. And so the same way a doctor can tell you have something funny on your kidney, until you biopsy it, you're not sure what you're looking at. You have hypotheses and you can constrain them, but the protocols are there to get to a conceptual site model that you know what you're looking at as you're dealing with these kind of dry cleaner sites. So I've got a couple of dogs here, and they're simulating bioactivity for me. And so when we inject bioremedies, essentially what we're doing is we're trying to be farmers, and we're trying to get stuff to go away. So the first thing we need to figure out is where the injection goes. And my 2 dog microbes there, they're quite happy. They're getting a carbon source. Imagine your injectate is some sort of nice dog biscuit that they like. And this is what my conceptual model of what injection looks like is that you put the injector down at a injection plane, and then the stuff starts squirting out from however it gets sent out. And the better we can control that to put the food where, hey, I want those dogs to eat my bad stuff, and I want them to marry up with each other and get the dogs where the food is, where the contaminants are, and I want to marry all that up so I can get this reaction to happen. Most times the microbes are just eating things, and it's wherever the remedy got shot, not necessarily where the contaminants are at. And so getting that marriage to happen, we need to know where that injection went, because those guys are just happy doing what they're doing. They're not particularly well trained. And on the second part is, do you get the bugs growing? And I used an example of either bread or cheese getting moldy. If you stick it in your fridge, you essentially have a cheese experiment going on under cold conditions. And if you wait a few years, eventually the cheese will degrade. But the start of that degradation, you'll get a little bit of mold growing on there. And if you had to predict scientifically a priori which spot of the cheese would initially get mold, super hard to do. It's biology going on, and they pick their spot from whatever conditions they did, but super hard to be predictive. It's just a moldy spot on the cheese, and where it happens at is fairly difficult to predict. And then as it grows, it's starting to consume the material that you're trying to get it to consume, but that pattern that is formed is biological, and it's based on variables that aren't particularly well constrained by stuff we're doing at the surface. And so determining whether they're happy enough to grow, whether they're growing in the right place, and what pattern they're growing in, and whether they're breaking down the thing you would like to break down, that's the thing we need to deal with. And so the simple statement of we're going to go farm some bioactivity is relatively simple. Getting the stuff in the right spot and getting the reaction to happen is actually fairly tricky, because we're asking things to be alive and go along with our program. So for watching injection, for this case study that we did, when we did that one, we learned a lot of things since then, and we've looked at a lot more injections. And so if we want to take pictures of injections, there's a couple ways we can do it. We can look for conductive material that you injected, or you can look for conductive areas that are bioactivity, or you can watch over time, and you can put an electrical well at the surface and watch changes. So in this case, we're looking at a 100-foot deep image, and we've got an electrical well at the surface, and then we got vertical PVC wells going down into the subsurface. And when you inject there along that injection plane, we get things that are not super surprising, but we get things that are not super useful in terms of injection. So if we look at where the water table is at, and then we think about what's going on, we've got a homogeneous sand aquifer, but when we inject a whole bunch of stuff and that purple signature went upward, and so we can think about that homogeneous sand, and we had a whole bunch of vadostone injectate. And then we have stuff that went down, and you get all this deep injectate that traveled 20 feet below the wells. So the wells saw material for a day and a half. The material actually was still sitting there, just in places that the wells couldn't see. And this effect of squirting around, we see that in all the injections we watch. And so the material goes out from that location, but it never goes out in any sort of really nice regular manner. It basically squirts out and goes places where it can. So in terms of going up, it went into the vadostone where it wasn't saturated. In terms of going down, that was a high K pathway in the, quote, homogeneous sand that was down there. And then if we think about bugs growing, typically we're thinking about getting conductive from bioactivity showing up, and it makes the subsurface more conductive. And it does it in a range. So in this case, we're looking at an increase in bulk conductance getting more purple. And what we're seeing there is it's got a funny pattern. And we're looking at, instead of looking at a vertical slice, we're looking at a map view of some data. And when we look at that, we see that the guys that are working with oxygen and nitrogen, they're a bit different in their conductance than the guys that are on the low end of the scale making methane and doing their things. And we can start seeing structures there, but there's also structures within that structure. And we see on sites where you'll have conductive tubes running hundreds of feet. And the biologists I've talked to, and I'm definitely not a biologist, but the biologists talk about them making cables to drop stuff off in different redox zones and play these different redox games. And they build these structures, and they're fairly complex on sites we look at. If you look at an old refinery or something, you get really bizarre conductive structures kind of built into the subsurface based on where they have carbon and what they're working with. So we have the simple version where conductive things equal microbes, but you have a whole lot more information about what's actually going on in terms of ecology. And this part of the research is called biogeophysics, and it's what does life do to things under the ground geophysically, and what does that mean so we can interpret it. So if you're going to go look at a moldy piece of cheese under the ground, this is a really moldy piece of cheese, and it's got species going on and it's got structure. So we can start thinking about what we're looking at, but we're on a research basis or as a scientific group of folks around the world, we're really far from understanding what mold on a cheese looks like at 50 feet. So for our case study, we have PCE in the ground. We have a former dry cleaner there in that little pink dashed area. And now it's a gas station. So we've got to think about how to investigate it, and we get some PCE locations. And so you got parts per billion concentrations of PCE, and then you've got monitoring wells installed down gradient, and you're watching what's going on. And then you have injection wells to try to remedy the issues. So fairly standard sort of approach, but when they did the injection, they didn't see the material show up in the monitoring wells, and so they wanted to understand where it went. So when they did that, we set up lines to look at, okay, we want to look between the injection zone and those monitoring wells that are down gradient and see what might have happened, and we go over the footprint of the dry cleaner, and we think about where to set up imagery such that we can see the parts we need to see for the conceptual model for the site. So understanding where the existing plume understanding is at, understanding features we want to see, and trying to capture that data. The red and the green on these lines represents the sides of the trapezoidal images we collect, and so we can think about where things are at and where we have full depth of image. And so you want full depth of image over the former dry cleaner pad in case there's a second source or something else you might encounter, and trying to think about how to deal with these things either in planning stages or in real time once you're collecting data out on the ground. This is an image of all the kinds of data that came out of that. So instead of having a few wells, each one of those panels is roughly 2,500 data points. So you've now collected tens of thousands of data points, and you're looking at, okay, what are we dealing with here, and what does this all mean? And so we need to think about this data set in the context of what we understand about how hydrogeology changes the ground electrically, and then what it means relative to the remediation objectives for the site. So if we look at one of these images with a simplified color scheme, there's some weird things about a dry cleaner that had an injection happen. And we see this on different dry cleaners around the world that we've looked at in terms of how these reactions happen and what kinds of things we're expecting. But we get in orange some really, really resistive stuff. And the levels of resistance in the subsurface are so high, it'd be as if you had some sort of a granitic intrusion with unfractured granite sitting there. But in this case, we're just looking at a sand aquifer. So that's definitely weird and strange. But we're also looking at PCE being in play, and PCE is a really good resistor. So if you put nice fresh PCE in the ground, you're going to get an excellent resistor. And so if you put it between the pore spaces, you'll get a nice dark spot where it's really resistive. And so that then becomes a target for us of going after those orange areas. The purple areas are way too conductive for this nice sand aquifer, and their structure is built around the injection system. So we go, okay, well, something else is going on, and we're expecting to see microbes growing. So when we're looking at these things, typically we're looking at things here below roughly 10 ohmeters, and that's where it tends to be a biological signature. We're also getting below 1 ohmeter, which tends to be sites where we have DNAPL degrading. There's some differences in the microbes, and we're still working on a biological basis what that is. And so there's folks doing lab work in biogeophysics to figure out what signals are created by what parts of their bodies and what reactions that they do. And there's actually fair level of debate as to why they can get that conductive, but that's a fairly regular thing for a solvent site that has degraders working on it. But if we go think about biopsying the data set, water table sitting there about 10 feet down, and that's also about where there's a slight change in lithology. But roughly it's just porous media with a silty sand over a sand. But if we then biopsy the resistor, we find PCE in there, and relative to DC, there's a little bit of DC, but the ratio is fairly high. It's mostly PCE, and there's no vinyl chloride. Those resistive places we sampled, the highest PCE we got was about 43,000. And if you recall, the original map was about 90. So we found some real hot spots, and they're not huge, so you get much higher concentrations, but the locations are much more constrained. And so we have hot spots around this domain, but they're not all over the domain, they're just spots. So that's an undegraded portion. If we go into the purple area, we still find PCE, we find DCE, and you go, well, maybe that's not biological, but there's also vinyl chloride present. And so we can see that in that conductive area, we got degradation ongoing. And we can think about, do we sample for particular dehalogenators? Do we sample for what else might be there chewing on the carbon that got put in the ground? And when we look at that, part of it's above the water table where dehalogenators not supposed to be happy. And we find that we've sampled dehalogenators in places that they're supposed to be incredibly unhappy, and they're still growing. So what does that structure mean, and what does it mean for sampling? And are we wasting some of the carbon, and do we need to spend more time making sure it stays below the water table? And so some of those farming issues are things that we don't normally think about enough in terms of if we're going to go farm some corn on the soil, we think about a soil sample, we think about the climate, we think about how much we're going to irrigate, and we think about all the parts to make that corn happy and growing where we want it to. But we also think about whether we're going to make a bunch of weeds grow next to it, and that's going to limit our corn growth. And so, you know, you can do things like spray herbicide, or you can think about how you work with your farming. So same thing down here is we put a bunch of carbon in the ground, we know we got some dehalogenator working, but are they slowed down by other guys that are happily working with the carbon? And so those ecology issues in the subsurface are things that I think we're still somewhat primitive with, and we can do things in the lab, but doing it in 3D in the subsurface out in the field, we're not that sophisticated yet. So you end up with a conceptual model where you've got confirmation that you got a biodegradation zone, and you got PCE there, and you got breakdown products all the way down through vinyl chloride. And then you've got undegraded locations where the injectate didn't reach and the material's functionally just sitting there. And so you then conceptualize of, okay, well, the bioremedy works, and now can we go treat these other areas and get them to be moldy as well? And how are we going to approach that, and what way do you want to tackle that? But you can think about that relative to, do we want to sample more with some wells in other locations that are looking the same as these locations, or do we want to take some more electrical pictures as we inject to see if we can get it to go to the right spot? And you're working through that logic electrically tied to biological samples and geochemical samples. If we slice the data and look at it in a map view, if we're in that vadose zone, the injection zone is there, the gradient is there, and that injection went upward into the vadose zone like we commonly see, and it coated that with carbon. And are those guys there? We know they're degrading PCE, but are most of those carbon atoms being used to do that, or is a bunch of that material being wasted to cause other guys to grow that aren't really helping our cause? Do we have a lot of weeds growing? And then down in the phreatic zone, we can see the injection went towards a chunk of PCE we were trying to treat, but it basically went around it for the most part. And so it went up and around a chunk you're worried about. And so you can then think about, well, let's just go target it directly and either heat it, eat it, or treat it, and whatever we want to do. But it says in this case, the biological approach is working, but we need to get it co-located with the areas that are a problem. And then there's also an area to the top of the image that you go, you got another chunk up there that you got to go deal with. So you can make solid models of these things in 3D. In terms of making them prettier, that helps for communicating between the different stakeholders on a site. But as visualization software gets better, it gets easier to port this stuff back and forth between different softwares and to utilize different tools that are being built. So as visualization software moves along, you can do a lot different things in terms of modeling what the biology is doing, modeling where it's hitting the various bits of geology, and what other redox zones you have going on. And it's easier to work with and come up with those images. A decade ago, it was a huge effort to get the basic 3D, but now the tools are getting better that you can start to play with the 3D a bit. So basic lessons learned, injections like to go up. And if you've got a nice vadose zone somewhere near your injection port, you're going to put material up there. When we look at solvent degradation, they're way more conductive than if we're looking at fuel degraders. And if we're looking at really old fuel degraders, we'll get them to get to that level of conductance with the reaction, but typically solvent degradation is happening at that really conductive level right at the start. So there's patterns there, and we can think about the ecology that's happening, but it has a habitat and a structure to that habitat. So it doesn't tend to care about fine-grained layers. It doesn't tend to care about, sometimes it's working with a flow path, but it could shoot across the flow path to go get something different in terms of a nutrient or a redox zone, and mapping that out, we can see where the structure goes to, but sampling enough to figure out why it wanted to do that, that's trickier. And it's like looking at the mold on your cheese in the fridge and going, why did it grow to the left side? And you're looking at whether the light bulb from the fridge was affecting something, or whether it sat next to different food on one side. But it's definitely a complicated pattern, and figuring out the whys behind life, it gets tricky. So figuring out the way the mold's working in the subsurface and be able to farm that, we need to get better at that. So in terms of moving forward, we've been putting in more electrical wells on different sites, both on a research basis and a commercial basis. And electrical wells look like a regular monitoring well, except for you pop it open and there's a cable, and it doesn't go vertically downward, it goes horizontally. And then you take one of those pictures, but you can watch it change over time and think about what's happening in the subsurface. And if you're trying to get remediation to occur, anything you do remediation-wise changes what's happening electrically in the subsurface, the same as it would change chemically or biologically, and you could sample that with a PVC tube. You could think about those electrical changes instead and be able to look at a broader range and be able to know where those changes are happening. But also thinking about how do we sample bioactivity? We can do total plate counts and you can do DNA, but which nutrients are limiting it, and what other things are going on there in the habitat. And I've looked at a lot of sites, and sites where we've tried to encourage things to grow where I'm watching, they won't grow. And sites where I'm like, oh, nothing should be growing there, there's lots of stuff growing there. And understanding which farm fields are really productive and which ones aren't, and what the real controls are. There's some things that are simple relative to pH and temperature, but there's some sites that don't care about these things. And there's some sites where the stuff grows great, and you're going, there's no reason why it should be happy here, but it seems to be much happier in some places than others. And every time I get hypotheses from different microbiologists about which things to test, the microbes do something that are violating the rules I'm getting. So learning those rules is going to take us a while, and it might take us 50 or 100 years as a group. And so we need to improve those farming techniques and get more efficient at how we farm. But using microbes, we're going to keep doing that. And so we've got to get better at how to locate them and figure out how to sample them to figure out how happy they are and whether they're going to grow in a particular direction that they would like them to grow. So if you get any questions, once again, we didn't harm any dogs, and contrary to one of my employees, we didn't put any cats in the talk either. So if you got questions, we're glad to answer them at this point.
Todd, we have one question so far. Is the webinar being recorded so that they can share it with coworkers?
Yes, and so you get a copy of that as a link, and so you'll get a CEU and a link to the webinar, and that's fully shareable, and you could do all those sorts of things. Technology is kind of fun these days.
Guy asked the question, your example was very instructive. Thank you. I imagine there are challenges at sites where the stratigraphy is much more complicated. For example, heterogeneous. How successful, hang on a second, long question. How successful has Aestus been in such tough sites?
So I call them my moldy sites, and seeing the conductive areas where microbes are growing is a fairly easy target. You're looking for something conductive, and electrical things are good at seeing conductors. The tricky part is if you took the mold and had a little bit on the sides of cheese, you can see the cheese, you can see the mold. It's once the mold covers the cheese and you're looking in the fridge going, there's something really moldy in the back of the fridge. Wonder what that used to be. Same thing in the subsurface. When we sampled that strong conductor in the dry cleaner, it gave us a really high hit of PCE, but that should stay about that conductance as it's getting rid of the PCE. So if you're in a resistive signature, you're something that's related to the amount of contaminant, and you can correlate to that. When you get moldy, you're related to what critters are growing there around the stuff that you're interested in for concentration. And so it's how to properly characterize moldy contaminated sites in terms of people want the concentration number, and you need to figure out how the mold is growing around it and what the center of that moldy area looks like. The other thing that we're finding for those kinds of sites is when they cable off and they build a big conductive structure out to the side, sometimes that's a flow path and it shows you exactly where all the stuff's flowing, and so you can then go find the channel because the microbes lit it up for you. But sometimes they're doing a reaction that they're quite happy with, and as people trying to clean up the site, we don't really care that you guys are working with sulfate. We want you to deal with this contaminant. But they're building out parts of their structure that are built for what they need, but aren't necessarily related directly to a contaminant concentration that you're worried about in the center of a plume. It's that they built off a structure relative to how they built the condo system.
Thanks, Todd. Neil Duran asks, can we hear more about the ongoing imaging using electrical wells?
So when you start taking differences of images, you've got all the QA/QC for an image, and then you've got that QA/QC again, and relative to the noise that you might create by collecting two slightly different images. And so we're looking at setting up some on a water supply aquifer with EPA of watching infiltration over time and watching the water table go up and down, the same as you would look at with a regular well. But we're doing that in a fractured carbonate system that's used for water supply. And so watch those processes you would normally think about watching with a PVC well, but watch it over a big chunk where you can watch where the fracture's at and where flow zones are at. We're also watching it in a faulted sandstone where there's microbes growing in the fault. And so we can then start thinking about how that habitat changes with rainfall. And so we're installing a horizontal well through there and watching the fault and then watching the microbes sort of bloom during rain events. And so we can then start thinking about when do we need to watch them, because as you start thinking about electrical well data having different time slices, you essentially have the shutter speed for a process, how fast you can take a picture, and that's on the hour scale. But then how often do you click the shutter again to capture events? And what we find is you can actually get events that cause more change than you can understand, and you need to speed up how often you take pictures. And so understanding how the ground is changing, it changes electrically a lot in certain situations, especially when biology is involved. And figuring out what the microbes are doing as they hit changes in their events, there's a lot of data there to chew on that we're going to be spending a lot of years figuring out what they mean. But for contaminated sites, we're figuring out we can watch site boundaries and we can watch remediation and watch whether something's going underneath a remediation wall or watch where the injection went. And so we're figuring out the protocols to do all those things.
Thanks. Jack Sheldon asks, Rush Boynton asks, how much did that cost, that example that you went through today?
So an imaging setup it's not a $10,000 thing and it's not a $1 million thing. It's like a $100,000 thing, and that can go up or down from that. But it's like that kind of a number because you're sending out a crew of people to go collect that data, and then you're putting it into a 3D visualization along with all the other data and integrating it. So it's about that kind of number, but it's thinking about, hey, I'm going to do remediation and I'm going to go spend about $1 million. Okay, we'll take 10% of that budget and figure out what you're remediating so that you do it once instead of injecting and then going, don't know if that worked, and trying it again or characterizing it again. And so it's very similar to if you take the dog to the vet and the vet says, okay, we did the blood test, might be something wrong with the kidney. Do you want the picture or do you want us to go pop open the kidney? And you go, well, let's take the picture and figure out what we're doing first and see if we need to do that. And that's a lot more costly when you first got to the vet. And so that same cost structure of we want to take the imagery and understand what we're looking at affects the oil industry, this affects vets and doctors, that affects us as well, as that cost is higher than an initial characterization, but a lot cheaper than a remediation.
And I think in today's dollars, the case study today would be roughly an $80,000-ish project from start to finish. Jack Sheldon asked similar question, can you provide a cost range for a survey assuming a dry cleaner or gas station size site? So I think you just answered that generically. Kirsten Marble asked, any suggested online resources for more information on this topic?
So there's the Aestus LLC website, aestusllc.com, and then we've got case studies and some papers and things, and so we can ship those out to you. And, so just ping us and we can send you up more stuff than you want to read.
Yes, we have lots of peer-reviewed papers and case studies, and depending on what the nature is of your site you might have in mind, we can focus our efforts to send you relevant information. Jack Sheldon also asks, can you conduct a survey like this proximate to an active pipeline?
Yeah, so we've actually going by a pipeline today. Most pipelines are coated, and so they're not great for showing up as a big conductor. And we've gone really close to newer pipelines. So if it's a really old pipeline and it's rusty, that'll get rid of being any sort of interference. And if it's a really new pipeline, we've got a site where they took a really old pipeline and had to replace the chunk that was leaking. And it was interesting because you had a weak signal from where the pipeline was old and it was rusty, and so we could get a little bit of a signal, but not much. And the new chunk disappeared because it was insulated. So we're familiar with going around those guys and working around them. And it's people looking at objectives of, are they trying to figure out where the leak went? Are they trying to figure out trying to demonstrate it didn't leak? And so working around those systems, typically they're not an interesting noise issue relative to what you might guess, saying, oh, I got a big metal pipe. Most times they're coated with rust or whatever coating they've got if they're newer, and it really doesn't cause much issue.
We actually just did a project in Texas where there was a crude oil leak out of a pipeline, leaked about 500,000 gallons. So we had to image around the existing pipelines, and it worked very well. Jim Cummings asked, no mention of biofilms, used just about every other term, structures and habitat. EPS seems relevant.
Yeah, so in terms of what's growing there, as I learn more biology, and I will still point out again, I'm not a biologist. As I learn more about it and I get to talk to microbiologists, they give me completely antithetical answers about what I'm looking at for these structures and these films, and whether they are microbes or whether they might be fungi or it's a combination. And so I think as we open the can of worms about watching life in the subsurface and trying to farm it, we're going to be learning all sorts of things that you go, oh, yeah, I guess that could be true. But I've been watching, there's videos online if you want to watch some fun videos. There's slime mold races where the slime molds go through mazes to go get the food and things. So these guys are not very large, but they do really cool things in the subsurface. So I tend to use very generic terms about what I'm looking at because I can sample them. I can take some DNA of water in those zones, but I know that I'm looking at something far more complicated than just bacteria or just fungi or different pieces and parts. And the signal we get people have hypothesized it's just the ions they're putting off, except for we're getting stronger signals than that. And then they look at whether it's just the biofilms, and they go, nope, not conductive enough. And then you look at whether they're precipitating metals or utilizing metals in their bodies. And there's a bunch of different things going on, but what that mapping of those signatures and what they physically relate to or biologically relate to is basically the great frontier of the subsurface that I think we're going to enter over the next 50 years.
Great, thank you. Other than Gu has a question about what's the typical ratio of lengths of the electrical line versus depth of investigation? What is the typical vertical and horizontal resolution? And Todd, I think I'll throw up our FAQ page for you.
Okay. So we're a 5-to-1 ratio. So if you're 100 feet at the surface, you're looking down 20 feet. If you're 1,000 feet, you're looking down 200 feet. You're not limited by technology, so we do 1,000-foot images on occasion. And it's thinking about that ratio and then thinking about a straight line that you can get somewhere in space. And we put lines through buildings and over water, and basically you can stick straight lines a lot of places on the earth, even though you go, well, it's an urban setting. That's usually where we're working. So that little figure up there shows the trapezoid getting bigger. Most of the sites we work at have a resolution on the meter scale. If we're working with, we've got a lot of customers that are looking at, hey, we've got a hard rock site and our water table's at 100 feet. If I get a drill rig out there, it's going to cost me $20,000 a hole to get down there. Let me take some pictures before I go down there. So you're looking at taking imagery to, 150, 200 feet, looking around the rock body, and then targeting where you're going to go biopsy. So in terms of that, your resolution, there's particular features that people go, well, is this going to affect my resolution? And we've gone through enough of these kinds of sites that we can tell you roughly what kinds of things you're going to see and what that target's going to look like, and if we need to modify something to improve the targeting. Any other questions, Stuart?
Yes. One last question from Russ Boynton. How would a high conductivity groundwater affect the results? For example, salty water near the ocean.
So we've looked at this kind of problem as like an ebullition area coming out into the ocean. And so it depends on what the seawater is doing and what your tide's doing, and you basically set up to go, okay, what's the question at hand? And if you're looking at like something coming out in the coastal area, oftentimes it's starting on shore. And so you work on the 3D to go, okay, I'm looking for a structure here. I'm not looking for a point in space. And so then you utilize the 2D planes to constrain what's going on and get a capture of what you're trying to look at going out off the coast. So it's the seawater is conductive, biology is conductive, but typically the structures that you get with them, you can see the difference in the structures and go, okay, this one is this conductor is going out into this other conductor. So you're just working with that, and you're looking at how to manage the contrast. Seawater is essentially as a bright light. Microbes in the subsurface are bright features, and so you're trying to manage the lighting basically of the data you're acquiring.
Thanks, Todd. That is all the questions. Oh, one more just came in, again from Rush. How long does it take to set up and run one of these lines to get a picture? Working in traffic area versus grass, etc.
Yeah, grass, you can get upwards of like 4 lines a day in an empty grass field. Typically in an urban setting, you're getting roughly 2 lines a day. If it's a lot of traffic control and lots of complexity relative to permitting and stopping traffic and doing different bits, you could be down to 1 line a day.
Great, thanks. Any other questions? And you have plenty of time to ask them. We'll be sending out a follow-up email with contact information. So if you have a question after the webinar that pops in your mind, please feel free to reach out to us. We're happy to answer them or discuss the site or whatever you need. We have one more just came in from Juliet Rose. How effective is this technology for heavier hydrocarbon compounds such as TPH diesel or creosote?
So those typically don't get as moldy. It's harder for things to eat those longer chain things, and so you'll get nice resistive bodies, and you'll get typically on those heavier things, the mold grows on one side, and so you'll get like a reaction pathway, and they'll be utilizing and kind of chewing on the side of it. So typically with those heavier things, you'll get resistive bodies with conductors sitting underneath them or like downgrading of them. And so that's how you're looking at those. And people ask me typically after I tell them what they're going to see, they go, how do I tell if it's mobile? And the picture on a single picture basis doesn't tell you that. It'd be interesting to try a temporal basis to see if you can see mobility issues in the temporal domain, but most commonly the biopsy, you go in and check that resistor for mobility and determine what you're working with. So we've worked with stuff that the biology is actually mobilizing it, and so you'll get a really resistive chunk that's pretty immobile, and that conductor on the side is guys chewing on it and mobilizing, and then you get this plume coming off of shorter chain hydrocarbons because they're breaking stuff down on one side of it. So it's a habitat and an ecology down there.
We've done quite a number of diesel sites very successfully, and also MGP sites and so forth. And I believe there's a couple of MGP site case studies on our website that you can access pretty readily. Any other questions before we close the webinar for today? Okay, well, thanks everybody for your time. We know it's incredibly valuable. There'll be a follow-up email coming out, as I mentioned. You may receive one or two emails. One of them will have the CEU credit information for your time invested today. And again, we're available for questions anytime. Very much appreciate it. I'll go ahead and close the webinar. Thank you very much.
Thanks, Stuart. Thanks, Todd.
Thanks, John. Thanks, Todd.